I'm very excited to present to you all our new magnetic Dynabeads-based ELISA products for use on the Kingfisher purification system. During this session, I will first cover the topic around Dynabeads-based ELISA, followed by automated bead-based ELISA. In the end, I will show you some results related to the assay performance and finally, clinical data. These automated Dynabeads-based ELISA products we have developed will capture and detect antibodies generated as a result of SARS-CoV-2 infection. The goal for all of us that work in the research field is to make everything we do in the lab count, driving the science fast forward with minimal technical mistakes. When developing automated bead-based solutions for isolation of immune cells, proteins, and smaller vesicles, such as intact SARS-CoV-2 virus and also exosomes, we took advantage of the very rapid binding kinetics offered by the Dynabeads to create fast protocols, while the Kingfisher platform made sure that we could handle large number of samples with high reproducibility, while spending as little hands-on time as possible with minimal error rates. It was therefore very tempting to transform the time-consuming manual plate-based ELISA, which may take several hours, and provide a high-speed, hands-off automated solution for detection of SARS-CoV-2 antibodies. One of the key features of Dynabeads used in any isolation protocol is the rapid binding kinetics. The proximity of the beads to the targets in the solution translates directly into short incubation times and therefore very fast protocols. This is also the case for these ELISA products. Only 10-minute incubation with a target is required, followed by 15-minute with HRP conjugated antibodies and 10 minutes with a substrate, with a few washing steps in between. And by putting the fast protocol onto the Kingfisher system, we have 45-minute walk-away solution for capture and analysis of SARS-CoV-2 antibodies. The hands-on activity is only limited to the initial preparation of the samples, Dynabeads and controls, starting by thawing and diluting the samples, resuspending the beads and prepare the control standards. Which control standards to prepare depends on the preferred assay. For a qualitative assay, all control standards are prepared and used to determine the presence or absence of spike antibodies. For a quantitative assay, an 8-point standard curve is created by making an 8-step dilution series from the high control standard. Serial dilutions of the sample may be required to ensure that the concentration falls within the dynamic range of the assay. The concentration of the unknown sample is then calculated using this standard curve. All the prepared reagents are then loaded into plates, and then the plates are loaded onto the Kingfisher instrument. In terms of dynamic range, standard curves for all three products have been created by plotting 8 target concentrations against the detection signal, demonstrating a strong correlation between target concentration and the detection signal, resulting in a dynamic range of up to 5,000 picogram per ml for the IgG, 4,000 picogram per ml for the total Ig, and 30,000 picogram per ml for the IgM antibody. These automated ELISA assays offers high precision. The reproducibility within the assay or intra-assay precision was established by spiking in known concentrations of IgG, total Ig or IgM, and analyzed in 6 replicates by one operator. Assay-to-assay reproducibility or inter-assay precision was established by spiking in known concentrations of IgG, total Ig or IgM, analyzed in 9 separate experiments by 3 operators over 3 days using 3 serum donors. High, medium, and low concentrations of target were analyzed, and data from the medium concentration is shown here. So both the intra-assay and inter-assay precision is very high for all these three products or three assays. Furthermore, two other parameters have been characterized, and that is recovery and detectability. Recovery was assessed by spike and recovery experiments, where a known amount of target was added to serum or plasma at different concentration levels, and the amount of each target was measured by bead-based ELISA. The data from the spike-in samples demonstrate that the recovery from the different concentrations are within the accepted industry standard, which is 80 to 120%, a range that sample matrix is considered to have minimal effect on the ability of the assay to accurately quantify the target. Detectability is presented as a signal to noise according to the ICH guidelines. The signal to noise was determined by comparing measured signals from spiked samples of IgG with those of blank samples. The lowest concentration, the green bars here, corresponding to 937.5 picogram per ml, are within the acceptable ratio of 2 to 1, which is the limit of detection, or LOD, and can be reliably detected by the Dynabead-based automated ELISA. The medium concentration, the red bars here, corresponding to 3,750 picogram per ml, are within the acceptable ratio of 10 to 1, which is the limit of quantification, or LOQ, and can be reliably quantified by the Dynabead-based automated ELISA. Finally, qualitative tests were performed on clinical samples to detect IgM and IgG. The data is presented as a ratio between absorbance values from the samples over absorbance values obtained by the calibrator. A ratio of less than 1 shows that the target is absent, while a ratio of more than 1.3 indicates that the target is present. A ratio between 1 and 1.3 is indeterminate and would require more testing. Clinical samples 3 and 9 are classified as IgM positive, while clinical samples 4 and 8 are identified as indeterminate. In contrast, clinical samples 1 and then 3 to 8 are classified as IgG positive using the same method. For comparison, a plate-based method is included. So with these products, we have transformed a time-consuming manual plate-based ELISA to a high-speed, hands-off, automated solution for detection of SARS-CoV-2 antibodies in serum or plasma.